I'm James Hadfield. I run the Genomics Core Facility at Cancer Research UK in Cambridge. It's a translational cancer research institute, and my lab runs the sequencing, the microarrays, not so many of those anymore, and the real-time PCR and other genomic technologies for the researchers in the institute. We're a core lab, and we're broad on the genomic platforms that we would offer, but the reality today is 90% of what we do comes off sequencing. I was primarily interested in Precise because of its simplicity. I mean, it looks a very simple assay to run. The molecular biology means we can go from RNA to something we can actually interrogate very quickly without too much messing around in the lab. Input requirements are good, and the throughput, it looks like we can run lots and lots of samples. And our experience with the very first plate that we've run is, it appears to be living up to what it says it can sort cells from very diverse populations, where we're doing large multicolour sorts in stem cell niches and such like. And to be able to do those in very high replicate numbers, sorting hundreds or thousands of cells into a single well plate, and doing multiple plates to look at hundreds of genes. Precise allows us to really scale up the size of experiments we do, rather than validating 10 or 20 genes, or maybe 48 genes on the biomark. We could comfortably look at 100 or so genes, and the numbers of samples we can process looks like it could be very, very much higher. I think the ability to potentially multiplex 12 plates of samples together in a single run, that would be a lot of qPCR by any other method. And I think that alone is looking like a very interesting development from a workflow perspective. I think it's the UMIs that certainly the research have, one of the first companies to put those into products, I think are going to be revolutionary for genomics.